Hello friends, today I will discuss with you another topic of anthropology. So far we have discussed so many topics of anthropology. These topics are all these topics are very important from UPSC mains point of view. I have discussed with you chromosomal and disorders and aberrations. What are like I have discussed with you what is uh, sex chromosomal anomalies like Down syndrome, like Edward syndrome, then Patau syndrome, Redo Chad syndrome. Okay, then I have discussed with you sex chromosomal abnormalities, Klinefelter syndrome, then Turner syndrome. I have discussed with you numerical and structural aberrations, how there is inversion of chromosomes, there is a deletion of chromosomes, there is a duplication of chromosomes, there is a ring formation in chromosomes. All these are due to lead to various types of genetic disorders. Some are autosomal chromosomal disorders, and some are sex chromosomal disorders. So what we do, we physical examination will find out disorders, as well as chromosomal and karyotype analysis, which through which we can understand chromosomal disorders, genetic disorders, and we can introduce various sort of gene therapy in order to we can introduce gene therapy to cure to treatment of disorders, as well as also we can do genetic counseling to of couples, so that whether they should go for a child or not, they both plan for child or not. So these are very much important. Sometimes there is a trisomy in chromosome number 21, that is called Klinefelter, that is called Down syndrome. And sometimes there is a chromosome number 18 gets trisomy. There are three chromosomes. It is called as Edward syndrome. And sometimes Patau syndrome, there is a chromosomal number 11 gets trisomy. So these are Cre2 syndrome, in which the chromosome number, some portion of chromosome number 5 is deleted. So it has to be discussed. So in order to know about the various autosomal aberrations and chromosomal aberrations, we have to discuss with you the various on the X chromosomes, on the sex chromosomal, like Klinefelter syndrome, when there is a extra X chromosome on the sex chromosome, and there is Klinefelter syndrome, and there is one X chromosome is missing in a female. It is called as Turner syndrome. Then comes super female. When there is a super female, we have trisomy. That is one extra chromosome on sex chromosomes. And what is what is intersex? We have one X chromosome. We have 46 46 that is intersex. We have to that they have a lot of things in common. I have discussed with you like what are chromosomal in orders. So there is a lot of things that are required to be discussed over here. Okay, so I have discussed with you a lot of things over here. Okay, then Klinefelter syndrome usually occurs in males. Okay, though what is there is a there is a extra X chromosomes in male, and Turner syndrome, जो है, it is usually occurs in females. Uh, there is a less chromosome. There is a XO, it is denoted by XO. Then there is a super female, it is occurs in females. Usually females are taller and have learning issues. And then clean filter syndrome, they have low testosterone level. Then Turner syndrome, it occurs in females. That is, there is a infertility in case of Turner syndrome. And there is a low level of testosterone in clean filter syndrome. And in Turner syndrome, there is a no fertility, no puberty, as well as. So this clean filter syndrome, Turner syndrome are usually intersex. Means they intersex means they do not people are not fit in either male or female categories. Okay, so that is called intersex. So yeah, for example, a person may have XY chromosomes but look a female outside. A person may have XX chromosomes but look male outside. So it is a natural variation. So they live healthy and they live normal lives. Okay, normal lives. Okay, then intersex is not a intersex. What is intersex? Is not a disorder. It is a variability. It is a natural variability. And many intersex people live healthy and normal lives. You all, I have discussed with you regarding these. Okay, I have discussed with you regarding super female. That is, female has one X has only one chromosome, and a girl is born with only one X chromosome instead of two. Okay, so clean filter syndrome, I have discussed with you that a boy is born with an extra X chromosomes. Okay, okay, it has a few female instructions in a male body. So uh, humans usually have 46 chromosomes, including. So let us break down clean filter syndrome, then Turner syndrome, then super female, then intersex. So humans usually have uh, 46 chromosomes. This means there are two sex chromosomes, and these sex chromosomes determine whether you will be male or female. If there are two X chromosomes, then it is a female. Then there is XY chromosome, then is called a female. So two sex chromosomes, there is a disorder in two X chromosomes. Then it leads to condition of a clean filter syndrome, Turner syndrome, and super female intersex. Then true uh, um, in XX, you can say that the person has a chromosomes of has chromosomes of a woman, but external genitals appears the female male. But in XY intersex means it means the person has chromosomes of Y, but external genitals are completely female in character. So this is XX intersection, this is XY intersex. So it is called, there are two X chromosomes, and when there is a problem in these sex chromosomes, then it leads to disorders like clean filter syndrome, like Turner syndrome, like super female intersex. Okay, XX intersex, XY intersex. Then comes we have people have low testosterone level, problem with using testosterone. Okay, so then comes true gonadal intersex means the it means that uh, XX persons have XX chromosomes or XY chromosomes or both. Okay, so it means varies the persons have both ovaries and testes. So it is called true gonadal intersex. True gonadal intersex. Okay, so complex or undermined intersex. So for example, 45 XO only one chromosome, and 47 means extra chromosome, either X or Y. So these orders do not result in a condition when there is a discrepancy between internal and external, and so complex or undermined intersection means when there is a condition of one X chromosomes missing or one extra chromosome. It is called intersex. So there is a one chromosomes that is missing, or one extra chromosomes. It's also the condition of Klinefelter syndrome, Turner syndrome, and it is also true gonadal intersex. So there is a I have discussed with you syndromic disorders. So there is a presence of one major and two major anomalies. So the definition of syndromic disorders is, I have discussed mosaicism and chimera. So there is a uh, mosaicism and this called chimera. So these are the two categories under syndromic disorders. So I have discussed with you autosomal aberrations, autosomal aberrations, and when there is a Down syndrome, autosomal abnormalities, sex abnormalities, then there is structural abnormalities, numerical abnormalities. There is a one chromosomes extra chromosomes in chromosome number 21. So Down syndrome, or then example though. Mm, so it had every chromosomes had a number of genes in it. So then I have discussed with you Patau syndrome. So this Patau syndrome is this uh, basically Advert syndrome. Advert syndrome is a trisomy 18, means the chromosome number 18 is present in triple. Okay, there are one extra chromosomes, three copies. They have three copies of chromosomes in each cell, in the body, instead of two usual copies. So there are two copies are present. There are 46 pairs. So out of it, the chromosomes number 18 has three copies. So it is called as Edward syndrome. Then comes Patau syndrome, in which the chromosomes number 13 has one extra copy of a chromosome. That is, there are three chromosomes. They are not one pair. The chromosome did not exist in pairs, but chromosomes, extra chromosomes is there in it. Okay, so then let us discuss about genetic imprints. What is genetic imprints? So I have discussed with you various lot of things like disorders and autosomal disorders, Klinefelter syndromes, chromosomic syndromes. I have discussed with you various types of mating: random mating, non-random mating, consanguineous mating, non-consanguineous mating, causes and changes which bring down gene frequency, like mutation, inbreeding. Okay, then migration, then isolation, mutation, inbreeding, isolation, migration, selection, breeding. Okay, then comes genetic drift. So then comes consanguineous and non-consanguineous mating. So there is a genetic effect of consanguineous and non-consanguineous. There is genetic effect of consanguineous and cousin mating. So there is a genetic load present in it. So there is a, I have discussed with you Mendelian inheritance. What is non-Mendelian inheritance? What is Mendelian inheritance? So Mendelian inheritance, single gene, single factor inheritance is an example of Mendelian inheritance. So there is a case of dominant allele and recessive allele, autosomal dominant, autosomal recessive. Okay, so sex linked inheritance, sex linked. So there is a presence of non example single gene disorders is are single gene disorders like Mendelian inheritance, like phenylketonuria, hemophilia A, hemophilia B, then Huntington's disease. Okay, so this is called as single gene disorders, Mendelian inheritance. Now non-Mendelian inheritance is inheritance patterns. So when there is a polygenic multi-factor inheritance are sometimes called as interchangeable. Okay, so there is a trait variation is thought to be caused by combined effect of multiple genes, not environmental contribution. So polygenic inheritance, multifactorial inheritance, polygenic, multigenic, or multifactorial inheritance. Okay, so there is a Mendelian inheritance. I have discussed with you non-Mendelian inheritance patterns. Okay, I have discussed with you Mendelian genetics. What is Mendelian genetics? So my conducted experiment gives two laws which follows Mendelian principles, that is law of segregation, law of dominance, and law of independent assortment. He conducted experiment on monohybrid crop experiment and dihybrid crops experiment. I have discussed with you various types of this pedigree analysis, then autosomal recessive, autosomal dominant, then comes twin study, co-twin study, biochemical method, then chromosomal and karyotype analysis, biochemical method, protein purification method, then electrophoresis method, immunological methods, DNA technology and recombinant technology, DNA technology and recombinant technology. So DNA technology and recombinant technology. So I have discussed with you a lot of things. I have discussed with you how chromosome. So there is a numerical aberrations, means there is a wrong number of chromosomes. When there is wrong number of chromosomes, not right number of chromosomes. Okay. I have also discussed with you what is autopolyploidy. What is polyploidy? Means polyploidy means what is diploidy? Means two set of chromosomes. Polyploidy means more than two sets of chromosomes. Means three sets or more sets of chromosomes. That is called polyploidy. Then monoploidy, polyploidy, diploidy. Diploidy means I have discussed with you what is diploidy. That is haploidy. Haploidy means when there are 23 chromosomes. That is when we talk about egg cell and sperm cell. So if they have one set of chromosomes, not two set of chromosomes, they have one set of chromosomes at 23 in human. That is called haploidy. So when these haploidy egg cell and sperm cell combine, the result is diploidy. Okay, so the cell has one set of chromosomes. The cell has one set monoploidy, so also called dihaploidy in some context. So monoploidy is one, there is a one set of chromosomes. Monoploidy, haploidy, diploidy. Okay, so there is a one copy, one set of chromosomes, two set of chromosomes. I have discussed with you. I have discussed with you structural aberrations, autosomal aberrations. Then I have discussed with you trisomy. Then I have discussed with you. Okay, a lot of things with you. I have discussed. So and what are mitochondria? I have discussed with you. Mitochondria are the power plants of our cell. They are the powerhouse of our cell. They take the food and they turn it into energy that our body can use. So it all happens in mitochondria. It also mitochondria, it are tiny structures inside most of our cells. So they take nutrients, mainly sugar and oxygen, and turn them into type of energy called adenosine triphosphate. Okay, so I have discussed with you other things also. I have discussed with you what is a degree of inbreeding, first degree of relation, second degree of relation, third degree of relationship. Okay, so I have discussed with you what is inbreeding, what is a degree of inbreeding. Okay, how the parents are closely related to which, that is inbreeding, that is no breeding, that is low inbreeding, that is moderate inbreeding, high inbreeding. Okay, so full siblings of parents offsprings mating, there is a mating between offsprings, there is mating between first cousins, there is mating between second cousins, totally unrelated, totally siblings, full siblings, or parent offspring mating, parent offspring mating. Okay, degree of inbreeding. So I have discussed with you inbreeding coefficient. What is inbreeding coefficient? So I have this inbreeding coefficient is probability that individual inherited two identical alleles for a gene, that is homozygous due to common ancestry. Due to common ancestry, due to common ancestry, they have the common ancestry. They have inherited. They have inherited F is equal to 1. That is no inbreeding, completely inbreeding. The coefficient in inbreeding coefficient means if there is a presence of a coefficient 1, then it means totally inbreeding. Then F is equal to 0 means no inbreeding. So it tells us about the level of breeding among the individuals at okay, the individual has inherited two identical alleles for a gene. It has identical two identical alleles for a gene. So one gene from parent side, one gene from the mother side, and they are identical. That is called inbreeding coefficient. Okay, so I have discussed with you in inheriting coefficient. I have discussed with you what is this autosomal dominant, what is autosomal recessive. I have discussed with you autosomal recessive disorders, autosomal sex-linked dominant and sex-linked recessive. I have discussed with you. I have discussed with you Mendelian population. Okay, what does a Mendelian do? Mendelian, how does Mendelian play? So the Mendelian population, they are there is a shared gene pool. They follow Mendelian population. Mendelian population is a group of interbreeding individuals that share a common set of gene, and they follow Mendelian inheritance law, and they share a common set of genes, common set of gene pools. Are interbreeding individuals. They are interbreeding individuals. Interbreeding individuals shared gene pool follow useful in population genetics. Okay, so Hardy-Weinberg equilibrium, Mendelian population, how low gene frequency behave in ideal population. I have discussed with you Mendelian genetics, this genetic polymorphism. I have also discussed with you what is genetic polymorphism. So it means genetic polymorphism means that people have not identical genes. People have different versions of a gene. People have slightly different versions of a gene. That is called genetic polymorphism. It is totally different to genetic polymorphism, Mendelian population. Then comes immunological methods in genetics. I have discussed with you. Okay, so they have one version makes blue eyes, one genes make blue eyes, make brown eyes. So there is a different versions of the same gene. So this is called genetic polymorphism. I have discussed with you various other examples like HBO is an example. HBO blood group system is an example of genetic polymorphism, and also this sexual dimorphism is also an example of genetic polymorphism. Genetic polymorphism is also very important concepts. Okay, guys. So genetic polymorphism. Uh, so let us discuss mm, genetic imprints. What is genetic imprints in human being, human disease? So usually I have told you that there are two copies of the gene. Okay, there are two copies of the gene. So I want to clear you one more doubt. Uh, human has 23 pairs of chromosomes. So one set 23 pairs comes from mother, and one set of 23 chromosomes comes from dad. So one copy is on the maternal chromosomes, and the another copy is on the paternal chromosomes. So these chromosomes in the pair are called homologous chromosomes. Okay, you have one copy of this gene on the chromosome number 5 from your mother. You have another copy of the same gene on the chromosome number 15 from your father. So same location means same loci, but on two different chromosomes in a pair. Okay, okay. So if both copies work normally, okay, if both copies work normally, sir, they are located on the different chromosomes, not on the two different copies are located on the different chromosomes. Okay, if one copy is the other may still work. This is called, if both copies are faulty, then it lead to genetic disorders. It case carrier, then one of the parent becomes carrier. Okay, so for example, chromosomes number 15, there is a gene there is a gene for eye color, and it is located in chromosome number 15. Okay, so you have one copy of gene on the chromosome number 15 from your mother, and you have another copy of the gene on the chromosome number 15. दो copies है, one from mother and one from father. Okay, so these two chromosomes in the pair are called homologous chromosomes. They are the same type. For example, chromosome 7 number, they are the same type. The chromosome number 7 from mother and chromosome number 7 from dad, but may carry different versions of the allele. But they may carry different versions of the allele. Okay, so they may carry. Okay, okay, I understand. The two copies of a gene are located. They are located on different pairs of chromosomes that you inherit. Chromosomes are inherited from your parents. So two copies of a gene are located on the same place, means they are located on two different chromosomes, same place on the two different chromosomes, especially on a pair of chromosomes you inherited from your parents. Okay. Then I have discussed with you that you get two copies of every gene, one from your father and one copy of gene from mother, one copy of gene comes from father. So what happens that one copy of gene is active, whereas the another copy of gene may be inactive. Okay, and other is turned off, so it is called genetic imprinting. Which one is active? So which copy of the gene, there are two copies, so which copy of the gene is active, it will depends upon the parents it came from. Okay, or if genetic imprint, if it is maternal imprinted, then means the copy from your mom is turned off. If there is paternal imprinted, it means dad's copy is off, there is no backup, and then can lead to problems. Okay, so that helps in brain development. Genes helps in brain development. The wrong copy is turned off, the working copy is deleted or mutated, then there is error in the display. When imprinting goes wrong, it can cause disease. Okay, so there is diseases called by imprinting errors. For example, Prader-Willi syndrome. I have discussed with you Prader-Willi syndrome. So what is genetic imprints? We will discuss genetic imprints. We will discuss genetic screening, genetic counseling, DNA profiling, gene mapping, genome study. Okay, we all will discuss in chapter number 9.4. Okay, so genetic imprints. What is genetic imprints? So people have two copies of genes, one from the mother and one from the father. So what happens, usually each of the two copies are active and turned on in cells. However, one copy of two is normally turned on. However, when copy is active on the parents of origin, some genes are normally active only when they are inherited from either person's father, or active only when inherited from a mother's side. So this phenomenon is known as genomic imprinting, genetic imprinting, maternal imprinting and paternal imprinting. There are two types. Okay, so there is diseases related with, I will discuss with you. First is Prader-Willi syndrome. It is caused when gene, one copy of gene came from father and it is turned off, and gene is located in chromosome number 15. So this is called Prader-Willi syndrome. Person is obese, intellectual disability, poor muscle tone. Okay, so poor muscle tone in Prader-Willi syndrome, obesity, small hands and feet, that is called Prader-Willi syndrome. Okay, so there is a problem in the chromosome number 15. Okay, then comes Angelman syndrome. So there are so many problems can come. Prader-Willi syndrome, usually when genes came from dad, it is turned off. And Angelman syndrome, what happens when gene from the mother is turned off in the same region. So there is problem of several developmental delays, Angelman syndrome. Angelman syndrome, there is mental retardation, speech impairment occurs in Angelman syndrome in the same chromosome number 15. So there are so many mechanism. There it is maybe due to deletion. 70% have a deletion of region of the maternal chromosome. There is a deletion. Prader-Willi syndrome, Angelman syndrome. Then comes back with Weidman syndrome. So there are back with Weidman syndrome. Okay, there are lot of symptoms associated with it. So it is very complex, not very well understood. So there may be uh, problem in the chromosome number 11. I have discussed with you uh, Angelman syndrome and Prader-Willi syndrome and Angelman syndrome. They have a problem with chromosome number 15. Then Beckwith syndrome, they have a problem with chromosome number 11. Beckwith-White syndrome. Okay. So there is a, then comes, there is risk of certain cancers. So they are caused by errors, imprinting errors on chromosome number 11. So genetic imprinting is like a switch that decides which parent's gene copy is used. It's like a switch, which parent's gene copy is used. It is because if the only active gene is faulty, if the only active gene is faulty, it can lead to genetic disease. These disorders are rare, but not understanding imprinting help doctors treat and sometimes treat them, diagnose and sometimes treat them. So genetic imprinting is very much important. Okay. Okay. Then there is Russell Silver syndrome. It is also a rare genetic condition that affects children growth after and before birth. They are shorter children than average. Okay. It is caused by a problem due to in genetic imprinting in chromosome number 7 or chromosome number 11. So there is chromosome number 5. Okay. So then there is problem with chromosome number 7. Okay. So these are imprinting disorders, not a change in the DNA, but in how genes are turned on off. Okay. So there are various problem. There is mental retardation. There is increase in the chances of cancer. There is a, low intelligence level. So genetic imprinting is very important topic from examination point of view. So there are usually disorders associated with it. So many disorders that are associated with it that I need to disclose. So there is delayed learning difficulties is also one such problem. Mental retardation, learning difficulties, motor skills, delayed motor skills is there. So they are Prader-Willi syndrome. There is a problem in chromosome number Prader-Willi syndrome. There is a problem in the chromosome number 15. In Angelman syndrome, there is a problem in the chromosome number 15. Then comes Beckwith Wides Beck, Beckwith Wideman syndrome. So Beckwith Wideman syndrome. So Beckwith Wideman syndrome, it is problem in the mental retardation, large tongue, body is fatty body. Okay, so Beckwith Beckwith Wideman syndrome is due to problem in the chromosome number 11, and Russell Silver syndrome, it is due to problem in the chromosome number 11. Russell Silver syndrome, motor skills. There are so many problems that delayed mental retardations. Okay, so so I have discussed with you a lot of things. Okay, Prader-Willi syndrome, we have Angelman syndrome, we have Russell Silver syndrome, Beckwith Wideman syndrome. So it is all due to genetic imprinting. So genetic imprinting is a rule in genetics. Okay, so here one copy of the gene is active, whereas the other copy of the gene is inactive, and it depends upon from where the gene came, either from the parents or from the mother, father or from the mother. We get two copies of every gene, and there are two copies. They are located on same location, same chromosomes pair, one from the paternal chromosome and maternal chromosome. So, but with imprinted genes, genetic imprinting, what happens is that one copy is turned off and other is turned off. So there are diseases associated with genetic imprintings. Okay, there are diseases related with genetic imprinting. Prader-Willi syndrome. It has been asked in previous year's questions. Angelman syndrome, Prader-Willi syndrome, Angelman syndrome, Beckwith-Wideman syndrome, and Russell-Silver syndrome. Then comes Prader-Willi syndrome. Then comes Angelman syndrome. Then Beckwith-Wideman syndrome and Russell-Silver syndrome. Russell-Silver syndrome. Okay. So next is genetic imprinting, and so all these things takes place during the early development of fertilization. So uniparental disomy takes place during the formation of gametes. Now let us discuss what is genetic screening, genetic counseling, genetic therapy. So genetic screening means we have to screen the population, and screening, what screening has to be done? There is a screening of genetic diseases in a group, in a population. They are either having the disease, or there is a probability that they will have the disease in future, or they will pass on potential to pass that genetic disease to offspring. So genetic screening is currently available for the following. So, so we do screening, we diagnose the parents with symptoms. So we can also advise other persons, okay, whether they have the disease, or if they, even if they don't have the symptoms, or they may be the carrier of the disease. Okay, so there are various types of screening. Okay, pre-symptomatic screening, career screening, paternal screening, newborn screening, forensic screening. There are various ethical concerns in it. Okay, so screening is done, genetic screening and genetic testing. They are directed at whole population or some portion of the subpopulation in which there is a risk of disease. Okay, so genetic screening is done for the whole population and genetic testing is done for the individual. And then after that comes genetic counseling. Genetic counseling, what we do, it is a communication process. It helps individuals, families, couples, and understand and adapt to the uh, implications of genetic condition, implications of genetic condition to the medical, reproductive, familial implications, psychological implications क्या होगा, we all discuss. So genetic counseling, who the parents or their relatives or family or couple are at risk of a disorder, they may be hereditary, they are advised about the consequences of the disorder and what is the probability that it will transmit it to their offspring. Okay, so genetic counseling, it usually it is a communication process and it deals with human problems. It deals with human problems that are, what is that problem? The party's problem is of occurrence of genetic disease, probability of occurrence of genetic disorder in a family. We all such things are discussed in it. Okay, so we do genetic counseling, we do genetic screening. So the purpose, but we do genetic screening, then we do genetic counseling. So the what is the implication, medical implication क्या होंगे, genetic implication क्या, what is the genetic implication, medical implication of the disease. Okay, these are guidance is given to them. Okay, then role of genetic counselor, help families, they do a lot of things. Okay, they, so there are tests are available to be done to detect the condition of disease. So what is the benefit? You can have the understanding of increased understanding that your child may it be born with Down syndrome, cystic syndrome, sickle cell anemia. They are communication process, and what is preventive measures, what are there, how to treat them. So better peace of mind, if the parents have a family history of such disorders, so it will provide some peace of mind when they come to know that they their offspring are not having any sort of disorders. It gives better peace of mind. It will understanding will increase. So it can be done early intervention. Maybe genetic testing, there is they may disclose genetic mutation that is hidden that may lead to development of deadly disease. How it can be helped by the counselor to let them prevent from developing or spreading. Appropriate testing are there. Okay, if your screening test, prenatal screening test, abnormal result gave, decision making, it helps them to understand the various testing options, various gene therapy are available, and what are the causes of such genetic disorders. They come to know and how it can be treatment can be done, how to have a better life out of it. All these things will be proved beneficial if genetic counseling is done. Okay, so I have discussed with you genetic counseling, genetic screening. So genetic screening, when we have like sickle cell anemia, cystic fibrosis, Huntington's disease, carriers of recessive genes. So to confirm, to confirm the patient is suffering with some genetic disorders or not. If the person is suffering from genetic disorders, then their counseling is are to be done by them, by the expert counselors. They will tell them about how to deal with such disease, what tests are available with them. It can advise family members whether offsprings will have such disease or not. It may be that there is some mutant genes hidden, whether it can lead to a fatal disease or not. Early type of screening, pre-symptomatic screening. So pre-symptomatic screening is when if a person have, if a family history is there of uh, or disorder. So pre-symptomatic screening can be done. If there is a family history of breast cancer, Huntington disease, okay, it may allow, it may help the patient to see whether there is problem with their body or not. Carrier screening means if whether the parents contain carrier genes. If they carry carrier genes, it is a possibility that they can, there may be a problem for their offspring. They are gene or chromosomal abnormalities can be discussed, can be done. So there are other types of screening that are there. That is parental screening, prenatal screening. So prenatal screening. So when there is a risk of the fetus may develop some genetic disorders, so it can be done blood sampling, serum sampling, ultrasounding, parental screening can be done. Newborn screening. Newborn screening can also be done with the when the baby is born with analysis of blood or tissue sample to detect genetic disease so that early steps can be taken to avert serious problem in future. So newborn screening, parental screening, prenatal screening, newborn screening, then forensic screening. So there is forensic screening can be done. So it can give linkages between the suspect and the evidence that are have been collected. Evidence and suspect. There has to be relation. There is a relation between them. Ethical concerns. There are a lot of ethical concerns in it. So I have discussed with you how genetic screening, genetic counseling, what is genetic screening, helps to screen the patients, is done at different stages, at prenatal stages, and uh, baby screening and forensic screening can be done. We can do genetic counseling. It is to communicate the probability of disease, whether the parents can, what is the probability that parents can pass their, uh, they are carry any genes, genetic disorders, abnormal genes, abnormalities. They can pass to their offsprings also. It can be help to take preventive measures so that it does not become fatal later on. Okay. So in most cases, I will want to say that they are not curable, but we can person's life can be improved, but they can be managed, they can be treatment can be done, but modern medicine improve it condition can be improved. So there is a genetic disorder is there is a mutation in the genes, either in a single gene, multiple gene, or whole chromosomes. So lot of things, there are therapies are available, hormonal treatments, medicines can reduce pain, gene therapy is there. I will discuss with you what is gene therapy. So most genetic is the condition caused by genetic disorders are not curable, but they can be some treatment can be given, they can be managed. There are various screening processes are available, genetic counseling are available. Now let us discuss gene therapy. So gene therapy means gene editing and gene therapy. What is gene editing and gene therapy? So means there is a problem in the genes, there is a problem in the whole chromosomes, abnormalities in the chromosomes in the one gene, multiple genes. I have discussed what are the diseases associated with genes, autosomal recessive, autosomal dominant, lot of things, genetics, we have discussed a lot of things. So means gene therapy means I want to change the genes in your body cells. So in order to treat or stop disease. So it can treat a wide range of disease, cancer, heart disease, diabetes, AIDS. Okay, it is an experimental technique by inserting a gene into patient cell. They don't use drugs or surgery, insert genes. So they are specific genetic disorders to focus. No different patterns of inheritance and targets of. So cell with there is a cell with defective genes, healthy genes are introduced, and cell functions restored properly. There are germline gene therapy. There are somatic gene therapy. Okay, somatic and germline gene therapy. So there is a question: can gene therapy cures genetic disorders? They can cure certain genetic disorders, but it's not available for all disorders. It is a developing field. It does not work permanently or effectively. It does not pump work permanently or perfectly. Gene therapy is like a fixing faulty genes, replacing faulty genes with healthy one, turning off the gene that cause harm. So if few genetic disorders have approved for gene therapy. It can cost, it is very costly, it is very expensive. They can cause immune reactions or complications. Long term effects are not known. Okay, so I will now tell you what is germline gene therapy and somatic gene therapy. In germline gene therapy, so somatic gene therapy, so somatic gene therapy, it is used for body cells like skin cells, blood cells, liver cells, etc. Affects only the treated person. In germline gene therapy, they passed on to the inherited person. Means it target cells is egg cell, sperm cell, or embryos. Ethical concerns are very high. Ethical concerns are very low. It is banned. Germline gene therapy is banned. So it is widely used in research and medicine. So germline gene therapy are only used in animals research. So but somatic gene therapy are used in humans for disease. So there is a lot of concerns. What is germline gene therapy? Germline gene therapy is used for animals only. Somatic gene therapy is used for humans also. Here the target is done on sperm cell or embryos. Body cells like blood cells, skin, liver cells, they are targeted. They ethical concerns are very high. They can be inherited, they cannot be inherited. So somatic gene therapy. And how there is a gene of interest, there is vector, and there is target site. There are three components of gene therapy. Target site means target cell, vehicle is a vector, and gene of interest. Gene of interest, vehicle, and target site. There are issues surrounding gene therapy. It has risk also. So I have discussed with you what is gene therapy. Okay, in vivo gene therapy and ex vivo. Now I will discuss with you ex vivo and in vivo gene therapy. Ex vivo therapy is done outside the body. In vivo means inside the body, and outside the body, inside the body. So what ex vivo gene therapy do? Doctors take cells out of your body, fix or add the correct gene in a lab, and then put the fixed cell back into your body. They take the cells, fix the cells, add the correct gene in your cells, and then put back the fixed cell into back in your body. In some cancer treatment, doctors take your immune cells, they reprogram them to fight cancers. In vivo gene therapy means doctors do the gene therapy inside your body using a harmless virus, a delivery vehicle that carries the gene. It is injected into the bloodstream and goes directly to the cells that need it. So there are in vivo, it is conducted in a lab. In vivo, directly inside the body, cells are fixed. For cancer, brain, muscle और liver disorders के लिए it is used. For blood or immune disorders, ex vivo is used. So there is a difference between ex vivo and in vivo gene therapy. I have discussed with you gene therapy, what we do in gene therapy. We do a lot of things in gene therapy. We do a lot of use. So gene therapy, what we do, we take new gene and insert into the body of the human being. Either there are two types of gene therapy, ex vivo and in vivo gene therapy. So by generally in ex vivo gene therapy, cell is taken from the body and gene is introduced into it, and so that the cell function properly. And in vivo, the cell, the gene is inserted with the help of vector into the body directly to the patient. So they are modified in vitro. Cells are again re-infused into the parent. It is very costly gene therapy. So then comes human DNA profiling, gene mapping, and genome study. Genetic screening. We have discussed genetic imprints in human disease. We have discussed genetic screening, genetic counseling. Now gene therapy I have discussed with you. Now I will discuss human DNA profiling, gene mapping, and genome study. Human DNA profiling, gene mapping, and genome study. Now what is human profiling? It is also called DNA fingerprinting. So it means the person is identified because of the unique DNA. Like your fingerprints are unique. DNA has also unique pattern. It can distinguish one person from the another person. So certain sections of the DNA. It is like super long barcode made of four letters A, T, C, G. So most of this DNA is same for all the human beings, but certain sections are vary between people. So these variable sections of DNA are used to create DNA profiling. How DNA profiling has to be done? So we have to collect a sample of blood, sample of saliva, sample of hair, or sample of skin. Extract the DNA. Look at specific areas of the DNA that are known to vary from person to person, like short tandem repeats. Short tandem repeats, they vary from one person to one. Compare the pattern of these repeats between the individuals. So compare. So this DNA profiling can be done. Each person has a unique set of short tandem repeats in DNA. So it is used in, we have to match DNA with suspect. This crime investigation to know the biological parent of a child, paternity testing, identify people from remains due to disaster victim. So I am 200% sure that a question will come on human DNA profile in this mains 2025. So missing persons DNA, okay, it is helps in studying human variation and ancestry. So it contains a lot of personal information. It must be handled carefully and ethically. The chances that two unrelated people have the same DNA is very extremely very small. It is like creating genetic iCard using your DNA. It is used in paternity testing, crime investigation, person identification, victim identification, missing person cases. Okay. 99.9% of human DNA are the same in every person. Okay. So the DNA fingerprinting was invented by Sir Alex Jeffrey at University of Leicester in 1985. Sir Alex Jeffrey. It is a forensic technique. It is a forensic technique. DNA fingerprinting developed by Sir Alex Jeffries. Okay. So there are certain patterns which are unique to individual. They are repeating sequences. So it was invented by Sir Alex Jeffrey. So how we can detect variations in DNA in the form of these mini satellites. The probability that having the same DNA fingerprint that are not identical twins is very small. So there are coding DNA and non-coding DNA. Some DNA codes for protein and some DNA are called non-coding DNA. So coding DNA are those DNA that gives instructions to make proteins. DNA that does not make proteins but may have other roles. That is called coding DNA. So only 1 to 2% of our DNA is coding DNA. 98 to 99% of our DNA is non-coding. Does not directly make proteins. But it is not useless. It has many important jobs. Okay. Coding DNA tells our body how to make insulin. Non-coding DNA around it helps control when, where, how much insulin is to be made. So coding DNA have was only 1% of the, but 99% of is non-DNA. Okay. It important, it controls genes activity. So what is mini satellites and micro satellites? Then I will discuss with you DNA profiling, what are ethical concerns, then comes DNA mapping, gene mapping, and then comes gene sequencing. Okay. Now, what are mini and micro satellites? So microsatellites and mini satellites, they look on our DNA. So they are short tandem repeats are called microsatellites. Variable number tandem repeats. They are long repeats. There are base pairs, 10 to 60 base pairs long. There is a lot of difference between microsatellites means there is sequence of letters, 2 to 6 letters. So they repeat a few to 50 times, found all over your DNA. Highly unique from person to person. So short tandem repeats is person to person different. So there is a sequence. So for example, like HI is being written many times. So microsatellites are used in DNA profiling, genetic disease research. It is a short sequence. It is a sequence of 2 to 6 letters. Mini satellites are very number tandem repeats. There are long sequence of 10 to 60 letters. They repeat dozen to hundreds of times. Then found near the ends of the chromosomes. So microsatellites and microsatellites. Short tandem random repeats, long variable number tandem repeats. So there are repetition is there of 2 to 6 base pairs. Repetition is there 10 to 60 base pairs. There are number of repeats, 5 repeats are there, or 50 repeats are there, or 10 or 100 repeats are there. It is used in DNA profiling, DNA fingerprinting. So why they are important? They are part of your non-coding DNA, highly variable between individuals. These short-term tandem repeats, long-term tandem repeats are very unique from individual to individual. So a lot of things have I've discussed with you. There are short-term tandem repeats. There are variable number tandem repeats. Repeats is of large number of bases. There are small number of bases. There is a it is one is short-term tandem repeats is short sequence. Variable number tandem repeats is a long sequence of base pairs. They both are used for identification. Okay, it helps our body. It's like a double helix. It has a double helix structure. It's like a twisted ladder. Those steps are made up of four letters: adenine, thymine, cytosine, and guanine. Always adenine always pairs with thymine, and cytosine always pairs with guanine. DNA, you have blood, DNA in your blood, hair, skin, and saliva. Even a tiny cell contains all your DNA. In the nucleus of almost every cell in our body, our half DNA is from mother and half our DNA from father. That is why you may look or act like both of them. What is DNA important? It performs sort of functions. It helps doctors to treat disorders. It used to identify people. It explains eye color, disease, health. It tells instructions how to stay healthy and function. It is just like your body blueprint or instruction manual. It is made of four letters in a twisted ladder. In a twisted ladder. It contains all the information to build you and run you. DNA is very much important. It stands for deoxyribonucleic acid. What does it do? It tells our body to grow. It builds our body. It grows our body. It tells our body to function. It tells our body to reproduce. It tells our body to look, how to look. DNA stands for deoxyribonucleic acid. Okay. So I have discussed with you genetic imprints in human disease, genetic screening, genetic counseling, human DNA profiling. Then I will discuss with you gene mapping. What is gene mapping? So what is gene mapping? It's like finding the genes, location of genes on a chromosome. Genes are located on a chromosomes. Chromosomes are present in the nucleus of the cell. We have to mark, just like we are dropping pins on a Google map to mark where things are. So here also marking is done. Genes have position are like houses along that road where each gene is located. How far the genes are from each other. Okay. Gene mapping important. Find genes, disease genes. Location of genes. How disease cause disease. It helps to understand inheritance. Develop treatments. Study gene function. Okay. So gene mapping. Location of genes. Distance between genes on So genetic mapping is technique from physical mapping method. So there are two types of mapping: physical mapping and linkage mapping. Genetic gene mapping and physical mapping. So I have discussed with you what is gene mapping. We have to locate the genes on the chromosomes where it is located, because it will help in detecting the disease. It will help in genetic therapy, gene therapy. There are two main types: linkage mapping and physical mapping. Physical mapping means measuring the physical distance between DNA, between the genes on the DNA. So we have to create gene map. It is expressed as the number of base pairs between them, DNA sequence. So physical mapping, then linkage mapping, how genes are inherited together. How genes are inherited together, that is linkage mapping and physical mapping. For example, a certain disease runs in a family and they keep seeing a specific DNA pattern with it. They can map that pattern to a region of chromosome and eventually pinpoint the exact gene causing the problem. Okay, this is called gene mapping. So restriction mapping, what is restriction mapping? Fish mapping and STS mapping. Fish mapping and STS mapping are used in gene mapping. Okay, so there is a physical mapping and there is S linkage mapping. Okay, so what is restriction mapping? It cuts, there are base pairs in it. There is a sequence, long sequence, short sequence, mini satellites, short term terminal repeats, long number variable repeats. So it cuts DNA into pieces to see specific sequences, like using scissors to cut a ribbon at mark point. No restriction mapping. Restriction mapping, then comes fish mapping. So fish mapping means restriction mappings, it means enzymes are used to cut DNA. We have used enzymes to find out the layout of a pieces of DNA. Okay. Fish mapping, fluorescent in situ hybridization. Fish mapping. So it is gene is located directly on a chromosome under a microscope. It is like a placing a glow in the dark sticker on a specific book. Then sequence tagged mapping. So it cut size in DNA. It Fish mapping is used to locate genes in chromosomes. STS mapping is where is landmarks in DNA. Use short DNA sequences. So I have discussed with you restriction mapping, fluorescent in situ hybridization, sequence tagged sites, STS mapping. Okay, so they use special enzymes cut DNA. Then is restriction mapping, fingerprint mapping, and optical mapping. Fish and STS mapping. This is physical mapping, and then is genetic mapping. There is a okay DNA sequence between genes. Okay, working out the number of base pairs between them. The fish, there are a lot of things that are required to be discussed over here. There are restriction mapping, fish mapping. It is like glowing to specific decants. It is, can be used under chromosomes to study. It is used to what is see in under a chromosomes. So to understand DNA sequence. Okay, and what is STS? Sequence tagged mapping. So just it uses sequences, short known sequences to understand the gene. It gives a detailed map. It draws a map. Okay. So restriction mapping, fish, STS are used in sequence tagged sites are used in mapping. Okay. So gene sequencing means exact sequence of bases A, C, G and T. Adenosine, cytosine, guanine, thymine, ACGT in a DNA molecule. The sequence of bases. Breaking the DNA into small pieces, assembling the sequence into single long consensus. That is gene sequencing. So what DNA consists of? It is made up of billions of nucleotides. They are like small blocks that build the whole structure. Each nucleotide has three parts, that is sugar. a sugar molecule called deoxyribose, then phosphate group. They link together the sugar and nitrogenous bases, a molecule that carries the genetic code. It forms the rungs of the ladder. So it consists of sugar, A always double helix structure, backbone of DNA, phosphate group bases carrying the genetic information, double helix, nucleotide, the building block of DNA, sugar, phosphate group, bases and double helix. It is a form of repeating blocks, nucleotides. This block contains a sugar, a phosphate group and bases. The order of these bases, it creates your genetic instruction. That is adenine always pair with thymine and cytosine always pairs with guanine. So there are millions of nucleotides and there are three parts of it, that is sugar, the phosphate group and the nitrogenous bases. So a phosphorus containing group, it links sugar together to form the sides of the ladder. Sugar means it forms the backbone of the DNA. A sugar molecule called deoxyribose. Deoxyribonucleic acid. Okay, it is like a double helix structure, three building blocks. Okay, so what are human genome project? मैपिंग की जाती है, सीक्वेंसिंग की जाती है, फंक्शनल एनालिसिस किया जाता है, डीएनए सीक्वेंसिंग की जाती है. ओके, व्हाट इज ह्यूमन genome project? It means mapping means doing genetic mapping, genetic sequencing, genetic screening, genetic counseling, complete set of DNA, all your genes, instructions needed to build and run your body. Humans have about 20,000 to 25,000 genes, and there are near about 3 billion base pairs of DNA. So global project, they involve scientists from UK, USA, Japan, Germany, France, coordinated by Department of Energy and US National Institutes of Health. Why it was important to understand genetics, genes, identify genes linked to genetic disorders, allow doctors to make medicine based upon disease, helps trace human origin and how are they related to other species. It was launched in 1990 and it was declared complete in 2023. In 2020, the finally gaps filled, 100% of the genome sequenced. Find genes, understand disease, create new treatments. That is Human Genome Project. It is very important. It was coordinated by the US National Institutes of Health and Department of Energy. So I have discussed with you lot of things like chapter 9.1, 9.2, 9.3, 9.4. I have discussed with you. Then we have 9.5. Then we have 9.6, 9.7, 9.8. Okay. So far we have discussed a lot of things. We have discussed genetic mapping, कैसे genes को, how we locate the genes, how we manage the genes, how we insert the genes, how DNA consists of. It is a three building blocks: a sugar, a phosphate group, and bases, nitrogenous bases. We have done gene mapping. We have done various techniques like short-term tandem repeats, variable number tandem repeats. They are short sequence, long sequence of bases, and they vary from person to person. So it is used in DNA profiling. Physical mapping is done in DNA profiling. Then we do genetic sequencing, do genetic mapping, location of genes on the chromosomes, human genome study. They all are interlinked. DNA profiling, gene mapping, gene sequencing, human genome project carried by US National Institute of Health and Department of Energy. So they are very much important from examination point of view. We have genetic imprints in human disease. Genetic, what are genetic imprints? Gene is turned off and on, and there are certain diseases related to it. Angelman syndrome, Prader-Willi syndrome, Beck with Weidman syndrome, Silver Russell Silver syndrome. So there is a defect in the chromosome number 15. There is a defect in the chromosome number 11 or 7. So genetic imprints. We do have two copies of genes, one from the parent mother and one from the father. So genetic imprinting works that one copy is turned on depending upon from where gene came, either from the mother or from the father. I have discussed with you genetic screening. Genetic screening is done to locate the genes, to identify the defected genes. Genetic counseling is done to guide the patients, to guide other family members regarding the probability of disease. Genetic mapping, we find the location of genes. There are short-term tandem repeats, long-term tandem repeats, genome study, human genome project. They all are interlinked. Okay. DNA is extracted, genes is extracted. So there are a lot of things that are discussed over here. We have to test the population to identify testing the population for a genetic disease, to identify subgroup of people that either have the disease or the potential of passing on it. We identify the people that are having disease and what is the probability that they will pass on it to the offspring. That is genetic screening. Then human DNA profiling, gene mapping, gene sequencing, location of genetic mapping. We do physical mapping, genetic mapping. In physical mapping, we do fish, we do STS mapping, we do restriction mapping. We used enzymes to cut the DNA into pieces. All these techniques are used in gene mapping. Then comes gene sequencing. Then comes genetic imprints, gene screening, genetic counseling, DNA profile. gene mapping and human genome project. So all these are very important topics. I have discussed with you various autosomal aberrations like Down syndrome, Edward syndrome, Patau syndrome, Cre-to-Chad syndrome. I have discussed with you sex chromosomal abnormalities, Klinefelter syndrome, Turner syndrome, super female, intersex, intersex XXY, 45, XX, 44, XY. They don't fall into male or female category. We have other syndromatic disorders like mosaicism and chimera. I have discussed with you in detail. What is a sex chromosomal abnormalities? There is a chromosomal abnormalities, extra X chromosome. That is a Klinefelter syndrome. So the chromosomes are, as we move from 1 to chromosome number pair 23, so its size goes on decreasing. So cells of the human body have additional X chromosomes that related to X chromosome. They have webbed neck, they have heart defects, kidney problems, motor skills development, mental retardation, webbed neck, Turner syndrome diagnosed. There are various steps for it. Numerical and structural aberrations. So sex chromosomal abnormalities, structural abnormalities are deletion, insertion, duplication, translocation, autosomal abnormalities, sex chromosomal abnormalities. I have discussed with you. Then comes numerical aberrations. There is alteration in the number of chromosomes. Numerical changes in chromosomes is called ploidy, euploidy and aneuploidy. Euploidy means normal chromosomes and aneuploidy, euploidy and aneuploidy. We have diploid, haploid. There is one set of chromosome, two set of chromosomes. Monosomy, one set of chromosomes. It usually occurs in eggs and sperms. So aneuploidy. Aneuploidy means euploidy, euploid and euploidy. Means euploidy means monoploidy, diploidy, polyploidy. Then hyperploidy, monoploidy, diploidy, polyploidy. Hypoploidy, hyperploidy, polyploidy. So there is condition of monosomy, trisomy, nullisomy, tetrasomy. Autopolyploidy and allopolyploidy. There are conditions associated with it. I have elaborated in a very simple and lucid manner. You have to go through the video in order to understand the topics in a detailed way, in an elaborated manner. Numerical aberrations, euploidy, aneuploidy. then euploidy, then aneuploidy, there are monoploidy and diploidy and polyploidy. Aneuploidy means hyperploidy, hypoploidy, monosomy, hyperploidy, trisomy, nullisomy, monosomy, nullisomy, and tetrasomy. So there is a missing chromosomes, extra chromosome is present there. The chromosomes exist in pairs. In normal somatic body cells, chromosomes exist in pair. This condition is diploidy. One set of chromosomes is called as monoploidy. The cell produces gametes or germ cells is containing half the number or somatic number of chromosomes. This condition is called haploidy. So hyperploidy, hypoploidy, euploidy, aneuploidy, autopolyploidy, and allopolyploidy. Then I have discussed with you four types of genetic disorders: single genetic inheritance, multi-factor genetic inheritance, chromosomal abnormalities, and then mitochondrial inheritance. So what is chromosomes and chromosomal aberrations? I have discussed with you. Then what is genetic effects of consanguineous and cousin mating, degree of inbreeding, coefficient of. So consanguineous and non-consanguineous mating, which brings out frequency. I have discussed with you genetic polymorphism, Mendelian population, Hardy-Weinberg law. There is a random mating. Gene frequency remains constant. Consanguineous mating and non-consanguineous mating. So genetic effect of consanguineous mating and cousin mating. Genetic load is there. I have discussed with you genetic polymorphism and selection, Mendelian population. There are four types of genetic disorders. Okay, mutation is there, isolation is there, migration is there, selection is there, inbreeding is there, and genetic drift is there. I have discussed with you Mendelian genetics in man family study, single factor, multi-factor, lethal, sublethal genes, polygenic inheritance in man. Then polygenic inheritance, then genetic polymorphism. I have discussed with you. I have discussed with you human genetics methods and application. I have discussed with you. There are so many diseases that I have discussed with you, like phenylketonuria, hemophilia A, hemophilia B, Huntington's disease, sickle cell anemia. There are so many diseases associated with it. Genes, all is all these things are because of genes. Chromosomal abnormalities. Genes provide instructions, like gene provide instruction for blue color, gene provides instruction for brown color of the eyes. ABO blood group system is an example of genetic polymorphisms. I have discussed with you chromosomal genetic disorders, genetic diseases, four types of genetic disorders. Inbreeding, I have discussed with you. Genetic load, I have discussed with you. What is genetic load? Means it discuss about the fitness of the individual. So consanguineous and non-consanguineous mating. first degree of relation, second degree of relation, third degree of relation. Genetic polymorphism I have discussed with you. Hardy-Weinberg law I have discussed with you. The genes remains constant generation after generation. Genetic variation in a population remains constant generation after generation. In the absence of allele frequency and genotype remain constant because they are in equilibrium. Genetic polymorphism and selection Mendelian population. Hardy-Weinberg law I have discussed with you. Mendelian genetics in man, family studies, single factor, multi-factor, lethal, sublethal, polygenic inheritance in man I have discussed with you. Lot of things. They are genes are made up of DNA. They tells how to make proteins. They fight germs, build your body, muscles. So there are about 20 to 25,000 genes. They are located in chromosomes inside the nucleus of your cells. They are instructions in DNA for making proteins. So part of DNA is called gene. The entire molecule that carries the genetic information is called DNA. Gene is much shorter. Thousands of DNA consists of billions of base pairs. So genes is a part of the DNA. DNA is a bigger thing, gene is a smaller thing. Gene is a part of DNA. So it contains instruction to build one specific protein. It contains all instructions to build and run an organization. It is a specific segment or it is a part of the DNA. The entire molecule that contains our genetic information. Gene is, if DNA is a giant book, gene is a one chapter in the book. Gene. Each chromosomes contain exactly one low continuous DNA molecule. It contains one DNA molecule. So chromosomes is a very long thread, and each thread is made up of one single DNA molecule. It is very long, tightly packed to fit inside the cell. Number of DNA molecules per chromosome is 1. Length of DNA in one chromosomes, it contains billions of millions of DNA base pairs. Number of chromosomes in human body 26. Number of DNA molecules in a 46. Basically chromosomes is a DNA molecule. 46 one per chromosome. Each chromosome is one huge DNA molecule. DNA is found out in chromosomes. Okay. DNA, chromosomes, genes, Mendel law, protein purification. Question is, do all genes Make proteins? No, all genes not make proteins. There are coding genes, non-coding genes. 1%, 2% of your DNA codes for proteins. Non-coding genes. So these genes do not, they make functional RNA molecules. Example, ribosomal RNA helps makes ribosomes, helps microRNA and long nodding RNA. Some genes make proteins, other genes make RNA molecules. That are important roles but don't become proteins. DNA is double, it is ribonucleic acids. It is a molecule similar to DNA, but it plays different role in your cells. It is in the nucleus and cytoplasm. RNA acts as the messenger and worker in the cell. It helps read DNA instructions and make proteins. It helps read. Messenger RNA, transfer RNA, ribosomal RNA, other RNA. DNA is the master blueprint. DNA or RNA are workers and machines in the factory. They help to build the final protein. There is messenger RNA. They carry the code from DNA to ribosomes, protein factories. They act as a worker, messenger RNA. They carry the genetic code from DNA to ribosomes. Then transfer RNA brings amino acids to ribosomes during protein assembly. Some brings ribosomes, some carry messenger code, transfer RNA, messenger RNA, tRNA, ribosomal RNA makes a part of the ribosome itself. Helps regulate genes and other functions. RNA. RNA. What is RNA? Okay, RNA. RNA, DNA, chromosomes. It is used the instructions. It brings carries the genetic code instructions to the ribosomes. It also contains in the ribosomes. It also carry instructions. It carry instructions from DNA to proteins. It helps assemble proteins in the cell. Messenger RNA, transfer RNA, write ingredients, amino acids to make the proteins. RNA is like a messenger and helper. It is work like messenger and helper to make the proteins in the cell. Without RNA, cell would not know how to build the things in it needs. Okay. So there is lot of things I have discussed with you like chapter 9.1, 9.2, 9.3, 9.4. So karyotype means photographs, chromosomal karyotype analysis, immunological methods, biochemical methods, cytogenetic methods are all the methods of genetic study. What is human genetics I have discussed with you. Deals with the principles of heredity and variations as observed in plants and animals and in man. So genetics is the branch of biology that deals with the law of principles of heredity and variation as seen in plants, animals and in man. It is genetics. Okay guys. Lot of things I have wonderfully explained things in my videos. Kindly go through my videos, will be enlightened to know about anthropology. Optional good score good marks in anthropology. Okay guys, channel को like करें, subscribe करें and share करें guys.